Wonderful. Hello, everybody. Welcome to Crossroads of Ideas, Growing Without Gravity. My name is Sam Mulrooney. I'm the Outreach Program Manager at the Wisconsin Institute for Discovery and also the Director of the Wisconsin Science Festival. We are happy to have you guys here tonight. I think it's going to be a wonderful evening, a wonderful conversation with our presenters. Real quick before we get started, just a couple quick housekeeping things. Crossroads of Ideas is a collaboration between the Wisconsin Institute for Discovery, the Morgridge Institute for Research, and UW-Madison Strategic Communications. It's an opportunity to foster dialogue between the community and UW-Madison researchers. The series addresses issues that matter to all of us and are also the subject of research at UW-Madison. Since 2014, Crossroads has been a staple of public programming offered at the Discovery Building at UW-Madison's campus. And before we introduce our presenters, I just want to take a moment to acknowledge the land that we occupy here at the Discovery Building. The University of Wisconsin-Madison occupies ancestral Ho-Chunk land, a place their nation has called Techob since time immemorial. In an 1832 treaty, the Ho-Chunk were forced to cede this territory. Decades of ethnic cleansing followed when both the federal and state governments tried repeatedly but unsuccessfully to forcibly remove the Ho-Chunk from Wisconsin. This history of colonization informs our shared future of collaboration and innovation. Today, UW-Madison and all of our presenting organizations respect the inherent sovereignty of the Ho-Chunk Nation and the 11 other First Nations of Wisconsin. Thank you. And tonight we are here to explore life's mysteries among the stars and delve into the groundbreaking research that is pushing the boundaries of our understanding of biology and chemistry and the unique environment of space. And we are honored to have as our moderator Dr. Eric Wilcots. Dr. Eric Wilcots is the Dean of the College of Letters and Science and the Mary C. Jacoby Professor of Astronomy at UW-Madison, where he has been the faculty member since 1995. A leader in advancing diversity and innovation, he launched the STEM Scholars Program, the Dean's Initiative for Diversity, Equity, and Inclusion, and oversees major campus projects like Levy Hall and the new CDIS facility. An expert in galaxy evolution, Dr. Wilcots earned his PhD in astronomy from the University of Washington and continues to lead the UW's involvement in the South African Large Telescope Project. Come on up.
I'll go over here for, this is live, great. So anyway, welcome, good evening, everybody. We're going to have a fireside chat, despite the fact that there is no fireside, but on a night like tonight, it feels like we should have a fireside chat. Roaring fire, Simon wanted a roaring fire, so we're going to get to that. But I'm going to be joined by my two guests here, who are going to join me on stage. First, let me introduce Dr. Simon Gilroy, who is a plant biologist here at UW-Madison in our Department of Botany. His research explores how plants and microbes adapt to spaceflight. He's a fellow of the American Association for the Advancement of Science. He serves on NASA's Biological and Physical Sciences Advisory Committee and is preparing his 7th plant experiment for the International Space Station. Dr. Gilroy earned his PhD in plant biochemistry from the University of Edinburgh and joined UW-Madison's Department of Botany in 2007. Simon, welcome. Why don't you come on up? So Simon is also the answer to a trivia question I like to ask my colleagues every once in a while, because as Sam mentioned, I'm a dean, so deans always thinking about money. That's what deans do. So I ask people, what department in LNS has the most money from NASA? And everybody assumes it's my home department of astronomy. It is in fact not astronomy, it is in fact botany, due to Simon Gilroy. So thank you, Simon, for doing that. Appreciate that. So joining us up here is Dr. Susanna Widicus Weaver, the Vaza Professor of Chemistry and Astronomy here at UW-Madison. She is an expert in prebiotic astrochemistry. We're going to get into that in a little bit, studying the chemical pathways that lead to life in interstellar environments. She combines laboratory spectroscopy, observational astronomy, and chemical modeling. In other words, she does it all to explore the mechanisms of interstellar chemistry. Before joining UW-Madison in 2020, she was a professor at Emory University and is now the provisional director of the Wisconsin Center for Origins Research. Susanna, come on up. So we're going to spend a little time. We're going to have a conversation up here, and then at the appropriate time, Sam will wave at me, and we'll open up for some audience questions towards the end. But welcome, both of you. Great to have you here. So Simon, we'll start with you. talk a little bit about what does it mean to put plants in the International Space Station?
It's a little bit more tricky than growing them on the ground. The ride is kind of tricky to get to. But actually, how plants grow in space, they grow well. So the biology, I always go, is an amazing force. Biology can adapt, and our terrestrial biology kind of works okay in space. It's not the optimal place for it to be, but humans, we work up there, plants grow up there. The kind of questions that we're interested in is, well, what does it mean when you take biology, which has existed on the surface of the planet and has always had that as its entire history, and move it to somewhere where it's never seen before? So what happens when you move, this is very dangerous with people who know what they're talking about. What does it mean? I'm going to now be very colloquial. When you get rid of gravity, and what does it mean when you move biology that's essentially been on the surface and then move it into an environment where the magnetic protection that you get from the magnetosphere goes down, and so radiation dosage goes up? Those are all the kind of questions that we think about. The practicalities of doing it is somehow you have to get your plant growth chamber with some seeds in it on the top of a rocket and get that rocket to go to the space station. Fortunately, the rocket and the space station we don't have to deal with. There's some very, very nice people who will do that for us. But all of the logistics of like how we're going to do it, that is kind of a very large part of what like space plant biology is. How are you really going to do the experiment? How are you going to get a plant to not be growing and then start growing when you put it on the space station?
As you say, you get rid of gravity, which we don't really get rid of, but we won't go into that. Yeah, in an extremely low gravity environment, what does change about plants and how they grow?
Yeah, so we're in the weightless environment of space, which is the classic thing that everyone has their vision of, like astronauts floating around and all that good stuff. And some of the things that happen when you move into that realm, where the 1xg that biology has relied on, we all are using at the moment, even though we don't think about it, gravity is totally dominating how you are sitting at the moment. When you start removing that, I always think there are things that you all are immediately thinking of, and it's very obvious. So think of an astronaut, think of us, that's an easy relatable. Your muscles start to disappear because you don't have to work against the force of gravity, your own weight. Your bones start to disappear for exactly the same reason, and you're not thinking about it at the moment, but your biology is monitoring the fact that you weigh something and is remodeling your bones constantly to that fundamental force which is there. And so remove those, and one of the astronauts, Don Pettit, Don just summarized it really nicely. Biology just gets lazy in space. And so what happens is your muscles disappear and your bones become less strong just because you don't need to maintain them. So take plants. Plants are going to be equally lazy. It's not going to be bones and muscles. It's going to be things like all of the strengthening tissues that you have, all the woody things that plants produce in order to keep themselves upright on Earth. Those are going to start to disappear. So those ones I think are very easy to think about. But then there are ones that, when you know about them, they make perfect sense. But I don't think they're the ones that are easy to guess. Somebody has to sort of introduce the ideas to you. And so the two ones which have a very big impact on how plants grow. One of them is that water now doesn't have gravity to pull it around. And if I just go, Eric, think of water flowing on the planet. Right? What are you thinking of?
Oh, a nice river, the water's flowing, waves at the ocean,
It's all
waterfalls, all that good stuff.
all driven by gravity.
Yes.
Yeah, all gravity pulling water around. That's what moves water at the macro scale for us. So if I take gravity out of the equation and put you in weightlessness, the forces that make water move and the forces that sort of dominate how it interacts with things become all of those molecular forces. So there's lots of hydrogen bonds, things like that at a molecular level. And so effectively what water becomes is very creepy. It wants to creep over surfaces by capillary forces, and it becomes very sticky. So it wants to stick to surfaces. So imagine that you're now going to water a plant. and you've got your watering can and you tip it on its side, well, nothing comes out, right? Because there's no gravity pull. So in spaceflight, we use pumps to move water around, and the classic pump is actually an astronaut with a syringe. Right? That's our general way of like adding water. Right? But so we're going to force water into the substrate that the plant's growing in. It's not going to be soil. We can talk about why soil is not a great way, but it's going to be a soil-like thing, like clay particles, something like that. You force water into it, but now, rather than if I had a pot full of soil and I pour water in it, gravity pulls the water down. That's gone, and so what happens is the water creepy and sticky, right? So it's going to stick to surfaces. It will stick to the particles of the matrix, the soil that you've got it growing in. It will also stick to the surface of the plant. And so sticky and creepy, it will want to start to creep over the surfaces of things. And so it will start to creep up over the plant. And if you add too much water, and that's like everyone, houseplants, right? You always overwater your houseplants. It's just like that's how we interact with plants. We kill them by putting too much water in them, right? In space, exactly the same thing. You add a little bit too much water, which is going to probably be the rule. The water will stick to the surface of the plant and creep up, and it will go up the roots and then up the stem and then over the leaves, and you'll end up with a plant that has a glove of water over the surface of it. And that's a very, very weird thing for a plant to be in, the condition, except that on Earth, you can totally mimic that. Just put the plant underwater, right? It's the same thing, it's flooded. So that is being flooded is just part and parcel of being in space as a plant. So it's this weird thing of like the physics is not what you are expecting. It's how physics works when low gravity, and it has huge impacts on biology. It has impacts on humans. Has impacts on plants. And there's these little bits where you go like, oh, I totally would have guessed that. And then things like sticky water. Who would have guessed that you get sticky water in space?
Well, we're going to get back to plants. Plants evolved from some prebiotic reality here on the Earth.
Yes.
What do we know about that?
So we know that early Earth was very hot. The water was not condensed. It was actually all steam. So early Earth was very volcanic, very, hot lava at the surface, not a lot of condensed water. Eventually, Earth started to cool. The water condensed out. We ended up with oceans. And it's from the oceans where life started to evolve. So we think the ingredients of life probably were incorporated into the planet as it was forming or delivered from comets and meteorites that hit it early on in Earth's history. And then from that, small microorganisms started to grow. And then from that, life as we know it now on Earth evolved.
It's such an easy picture. Talk about how your science tries to untangle that and test that.
There are many ideas about how that evolution of life might have occurred, and we have not yet figured out any one particular one that worked all the time. We think it's probably some combination of ices and warm little ponds, as Darwin would have said, and single-celled organisms being trapped in an area where they evolve into something more complex and things coming in from space. So the part that I study is actually how those ingredients as the planets form are formed in space, and how does that material get processed as you go through star formation and then planet formation. So if we look at other solar systems like ours, is there an Earth 2.0 out there? We don't know yet, but we're trying to find it. Colleagues and I are actually really actively trying to find Earth 2.0 right now. But one of the ways in which we go about this is we look at the chemistry of these regions in space where other sun-like stars are forming, and we try to understand what might have been available as our solar system was forming and what sort of chemistry you would expect to see on those planets that we find around other stars. And then from that, try to make a guess as to whether they could be habitable, whether there could be life on those planets. And someday, hopefully, we'll be able to tell whether or not we're right.
You say you and your colleagues are trying to find Earth 2.0.
Earth 2.0.
How do you do that?
So to do that, you use telescopes to watch planets either pass in front of their star and you take a spectrum of the atmosphere, or we look at the gravitational pull of a planet on the star and we can see the star wobble. And so from that, we can tell that there's a planet there. And then we know based on what we understand of solar systems, based on the star and how far away that planet is and how big that planet is, whether or not there could be liquid water at the surface. If there can be liquid water at the surface, there's a chance for life. So that's the definition of habitability as we define it right now in the field. If we ever find a way to make life happen in something other than water, then maybe that opens up the possibility. But for now, the big assumption in the field is that liquid water has to be at the surface of the planet for there to be life on that planet. And so then we start to collect information about what other molecules are there. So we track the chemistry, and we try to find things that are in the atmosphere that cannot be produced through non-biological processes. So if geology can result in that molecule, that is not something that will tell us if there's life there. If standard weather processing can produce that molecule, or if that molecule can be assumed to have been there when that planet formed, those things are not what we call biosignatures. So we're looking for molecules that tell us very specifically that there's life present on that planet. And one way to do that is to see if it's out of equilibrium. So we know what it should look like if there's no life. And so if life is there, it throws the whole system out of equilibrium. And so we have a very clear set of signatures on Earth as to what Earth life does to our atmosphere. And we're trying to figure out if that is universal.
So you've talked about habitable planets. Earth is habitable.
Earth is
Let's
habitable.
make sure we all understand where we are in the solar system. Let's get our scale set here. Where are we in the solar system?
So there are the rocky inner planets of the solar system. Earth happens to be in just the right area to be warm enough to have liquid water at the surface, but not too hot to evaporate all the water. So Venus is too hot. Venus is in closer to the sun. Mars is a little too cold, all the water's frozen, and there's not much of it. And so Earth is in that Goldilocks region. We call it the habitable zone. That moves depending on how big the star is, how bright the star is, what age the star is. And so that habitable zone will be different in different solar systems. Our solar system has one habitable planet and two that are on the edges. Other solar systems have many habitable planets. Something that's interesting, though, about our solar system, if you go to the outer solar system where things get very cold and you have the big gas giants, so Jupiter, Saturn, Uranus, Neptune, if you take a look at the moons of those gas giants, there are many moons in our solar system that actually have water at their surface. And so it may be that habitable moons are more prevalent in the universe than habitable planets, but moons are very hard to detect. So first we have to find the planets, and then we'll go for the moons someday.
Got it. Okay. So we're in a habitable zone. A couple planets on the edge of the habitable zone. Let's have a little fun. We're going to talk about one of those planets on the edge of the habitable zone, Mars. How long does it take to get to Mars?
Are you walking? Or
Presumably not.
yeah, this is the bit that I think is just very, very hard for us to grasp. Space is big.
It looks like it's on.
All right, how about
There's a
that?
light. There's a light.
There you go.
Go.
All right. Goes off when it goes in my pocket. All right, how about the, that's good.
Well, yeah.
Yeah. Space is big. And at our scale, it's very difficult to sort of comprehend how big space is. So rockets, right? Rockets go incredibly fast, something like 25,000 miles an hour. Get a speeding ticket, but you don't care because the cop can never get you. Right? You're just going incredibly fast. But to put into an idea about how far we're talking about. All right, So imagine you're on a rocket, right? Now, everyone blink, all right? Everyone blinked. This is going to feel like magic. You would be at the state capitol now, right? Just in the time it takes you to blink, you would have gone to the Capitol. In the time that it's taken me to tell you about that, just this amazing, I'm stringing this out because I'm trying to work out how long I've been talking. From the beginning of this sentence to the point of where I'm telling you how far everything is, right? If you were on a rocket just about now, you'd be in Chicago, right? It's just, so rockets go incredibly fast, but the distances are just incomprehensibly large. So to go to Mars on a rocket, the fastest trip that you'll make is about 9 months to get there. All right? So a blink of an eye, you're at the state capitol, you're going to be sitting on that rocket for 9 months. So you imagine now the logistics of a crewed mission. And this is one of the big goals. Is there's a great thing in NASA, bots before boots. All right? Work out how to do it with robots, work out how to find out what's going on, send rovers, do all of that. That's an incredibly important part of it. But the reality is, we're human beings. We want somebody wandering around on there. We want the Martian in real life. So a 9-month trip, once you start going to those kind of distances, then you're talking about, you know, if I want to talk to you, I can say something, but if you're on Mars, you've got to wait 20 minutes for me to be able to talk back. So logistically, things get incredibly complicated. And the reality is that the going to the moon, you know, a few days to get there, that's a camping trip. Going to Mars, you have to be self-sufficient. The distances are just so enormous. And so if you're going to go to Mars and you're going to be self-sufficient, one thing should probably be a good idea. Try and feed yourself. Got to be self-sufficient. You could fly enormous numbers of spacecraft carrying the world's supply of Twinkies and dump them on Mars. And that would be one way to imagine self-sufficiency. But that's both incredibly expensive and it's not really sustainable. It just costs so much and it's so tricky to do. And so that's one of the goals for like the Plants in Space program is the way that we are self-sufficient on Earth is we grow our food. And the question is, can you take that model and then move it to this planet kind of in the habitable part, right? You can't go outdoors and plant your plants, but it's benign enough that you could imagine that maybe we could kind of move Earth to Mars. But it's not going to be Earth 2.0 because Mars, hugely, hugely different.
So it's going to take a while. We got to bring our own plants.
Yep.
So if you're going to Mars, bring some plants.
Yeah.
That's lesson number 1.
Yeah.
Susanna, we have sent a number of bots to Mars.
Yes, we have.
Part of their mission is to determine whether life ever existed on Mars. Where does that stand right now?
We see signs of previous activity on the surface of Mars with liquid water. So at some point, Mars had water at the surface. At the moment, it's frozen out. So if you want to have access to water, you probably have to dig down below the surface where it's a little bit warmer. We have not yet found definitive proof of life on Mars. There have been groups that have claimed potentially fossilized structures of bacteria in Martian meteorites. That was a big deal when I was in college, and then it got refuted, and it's been back and forth several times. They have not yet found anything that is a clear-cut case of microbial life on Mars, but they've seen signs maybe. So there is that possibility that at one point in time at the surface of Mars there was some sort of life, but we don't have any evidence that tells us for certain that that's true. So these rovers are actually going and taking samples and taking measurements, looking at the chemistry of the soil at the surface of Mars and trying to understand what ingredients might be there and to continue to look for potential signs of life. But there haven't been any discoveries.
Nothing yet.
Not yet.
But when we go there with our plants and dig around,
I would bet you almost guaranteed to be able to find microbial life on Mars. Go and look where the rovers have been.
Yes.
Because microbes, So we're going to send a rover to Mars, and planetary protection is a big deal in NASA. And planetary protection used to be the bugs are going to come and kill us. And it was, when the Apollo astronauts went to the moon, it was an incredible amount of care bringing samples back from the moon because, they were going to have some moon virus or something like that. And it was going to be damaging to Earth's biology and potentially catastrophic. And then, very quickly, it was very clear that that's not true. And so planetary protection now is much more an outward-looking thing. And we don't want to screw up the rest of the solar system with our biology and then go and ask, well, are there other life out there? Because then you're guaranteed to find it, because we put it there. So planetary protection and things like rovers are intermixed, and they're incredibly careful about decontaminating it, and the clean rooms are amazing, and they send these things off, and everyone is pretty happy. There are microbes on them, just because microbial biology is so resistant. The idea that you could really get rid of it on something as big as like a Volkswagen van, which is what these rovers are like, is just unrealistic. So I'm pretty certain there's life on Mars. It's just that we put it there.
We put it there.
Well, that's encouraging. Thank you. You're making us feel better.
But look somewhere else. Don't look where the rovers went. Look somewhere else.
That's okay. Okay. All right. You mentioned, and I'm going to ask this to both of you, radiation. Right, space will try to kill you in lots of different ways.
Yep.
Talk about the challenges of radiation, both from sort of the space flight perspective, but also as you think about early biology in a radiation-rich environment. How does that factor into your thinking about the rise of life?
We can go the beginnings of life.
Okay.
Dying to life now dying, or we can get, let's do
Okay, so actually the radiation helps us out in the beginning. So we need the radiation from the star as a new solar system is forming to cause the chemistry that leads to the ingredients of life. So without that radiation driving bonds breaking and reforming, we would never build up biochemistry. So we would never get things like amino acids and sugars that are the ingredients of life as we know it here on Earth if we didn't have radiation causing that chemistry to happen. Once you get to an Earth-like planet, again, we need that radiation initially to cause that chemistry to take off. So if you have very simple molecules in the ocean, that radiation penetrating the water actually causes that chemistry to form more and more complex molecules, and that drives that chemistry. But once we get to the point where there's a life form, then there has to be some sort of way to shield that life form from that radiation, or in the same way that it breaks down molecules, it'll break down the life form. And so we have to have a balance there between what we need to build up the life in the first place and what is present that could damage the life once it forms. So I guess that's where I hand off to you.
Radiation bad. That's a good place to start with. All of us are being irradiated as we're just sitting here. Right? There's a background radiation on the Earth, but to put it into context, to give you a feel for what it means to do a space flight away from the Earth, and we're going to say that we're going to have to move into deep space, away from the Earth's magnetic field, because that is protecting us. We can talk a little bit about that in a minute. So you're going to take this trip to Mars, right? And you're going to have a 9-month trip out there. And very fortunately, when the rovers were sent out, for one of them, they switched on the radiation dosage meter during the flight out. So we absolutely know what the kind of radiation profile there is on that 9-month trip. So you're sitting on the Earth now, you're being irradiated by background radiations coming out of the geology of the planet with radon gas and things like that. This sounds scary, right? And there's cosmic radiation coming in, but we are incredibly highly protected by the Earth. And so, none of you, hopefully, are really freaking out about this background radiation dosage that you're getting. It's just part of being alive, right? So let's assume we're all going to live to be 90 years old, right? That's a good aspiration. I'm going to live forever, but let's assume that we're going to set a goal for 90, right? And so you've got this low level of background radiation that you're going to accumulate over 90 years, your entire lifetime. When you make that 9-month trip to Mars, you get it all in one go. So the 9-month radiation dosage is your lifetime's background radiation dosage on Earth. And that, again, might feel really scary. It's not. I always go, you know, the movies, it's like, radio, like rip your face off radiation. It's nothing like that. It is a constant, energetic radiation, but at a relatively constant low dose, but much higher than you get when you're on the surface of the Earth. And so, and radiation, when it hits complicated biological molecules, does chemistry to them. And so, you know, we all hear about radiation and damaging DNA and cancer, but it'll do a number on all biological molecules. So, you know, a cosmic ray will hit a water molecule. We're mostly water, right? So it's a pretty good chance it'll do that. It will cause chemistry in that water molecule, and what's the products of that will react with the biology around them, right? And so it's going to be damaging. It's not going to torch the biology. Biology is incredibly resistant, and we have mechanisms to repair that kind of damage. But once you go into space, the radiation dosage goes up considerably, and it is going to start to degrade all biology. And the Earth's magnetic field is protecting us from a lot of that radiation dosage, and we're going to go outside the Earth's magnetic field. And we talk about Mars. Mars doesn't even have that protection for you, right? So the Earth is I like it. I think we should keep it. It's a great place to be. The radiation dosage is going to be high. It's going to degrade biology. Microbes are fantastically resistant a lot of microbes are fantastically resistant to radiation. Plants, pretty resistant, about 50 times more resistant than human beings. We are the weakest link, right? And that might be that we're just such a complicated piece of biology that it doesn't take much to really screw up how we work. So yeah, and there are things called showstoppers within NASA. And a showstopper is a challenge to human biology, because we're thinking about the crew now, a challenge to human biology, which means that humans can't exist for long times off the surface of the planet, and there is no countermeasure that we have yet come up with. So there's no piece of technology, there's no shield, there's no biological countermeasure. And deep space radiation might be a showstopper. It might be that, you know, the Earth is so protective of us that I really don't think this is going to be true, but it could be that if we go for long periods of time into deep space, the radiation environment you're in is just going to be the showstopper. But I have, you know, great technology. Biology is incredibly resilient, and there are lots of biological countermeasures that you can come up with. And so the combination of those is, I think, is going to be a way of getting around this. But radiation is a really big deal.
Wow. Anybody feeling good now? Since we're talking about Mars, The Martian.
Yes. Take the potatoes.
How many of you have seen The Martian? Read The Martian? Yeah. Enough of you. On a scale of this is complete utter fantasy on one side of the scale to, wow, how did they get it so realistic on the other side of the scale? The Martian.
Do you want to go with that? I have my number.
Oh, are we supposed to come up with a number?
Yeah,
1 to 10?
it doesn't mean a number. It can be. I didn't give you a number scale.
I'd say maybe 6 or 7.
Leaning which
Towards realistic.
Okay,
The dust storm at the beginning was total fabrication.
Yeah.
Mars doesn't have enough of an atmosphere for that to happen. So that was like the driver of the whole story, right? The dust storm destroyed everything. But the rest of it, actually, the engineering and some of the science, I don't know about the plants. That's not my area. But in terms of the engineering that it took to get him off of Mars, I think it was fairly reasonable.
Okay.
Yeah. I'm just assuming I came in late and didn't see the dust storm, because I agree. So that was like that is like, you can do the calculation of how fast the winds were, and they're just thousands and thousands of miles an hour. I would give it an 8, a good solid 8 towards the high end of 8. Yeah, it's good.
Right. You've heard it here, folks. You heard it here. You mentioned a couple other exciting things in the solar system, Right. Moons. I'm thinking about Titan, Saturn's largest moon, and I'm thinking about Europa.
Europa's been in the news a lot.
Europa's been in the news. Why don't you tell us a little bit about Europa, why it's in the news?
So Europa is our best chance of finding liquid water at the surface of another body in our solar system. Europa is a moon of Jupiter, and we have just launched the Europa Clipper mission to actually visit Europa and take measurements of how much liquid water is at the surface, how deep is the ocean, and some very preliminary chemical measurements to see what else might be there, and a lot of other measurements just to get kind of a physical diagnostic of the moon. But when we launched the most recent Mars rover, I had a Facebook discussion with many of my NASA friends about if you were going to get space craned onto an object in the solar system, what would you choose? And by far, Europa was the #1 answer. We all want a submarine to just plop us right there. So Europa's very exciting because I think Europa's our best chance of finding life in our solar system outside of Earth. Titan is an interesting option. Titan's chemistry is weird. The lakes are methane. So if there's life on Titan, it does not really look like life as we know it here on Earth. But there's a mission coming up to go to Titan. It's called Dragonfly. They're building a quadcopter with a bunch of instruments on it, and they're going to drop it in the atmosphere, and it will fly, sample the atmosphere, land, sample the soil, sample the lakes. So we'll learn a lot about Titan in the next 2 years. So I think the target launch date for Dragonfly is 2027. So we have a lot of solar system exploration going on with these interesting moons. And then there's also Enceladus, which is next door to Titan. Enceladus has an ice crust, and it has tiger stripes where the crust has cracked. And there are geysers of water that blow out of that crust and very far above the surface of Enceladus. And actually, some of the material from Enceladus rains down on the surface, on the top of Titan's atmosphere. So there's a chemical link between the two. So we will learn a little bit about Enceladus when we go visit Titan again as well.
Awesome.
So it's an exciting time in planetary science.
So Simon, have you or your colleagues thought about plants in a subsurface ocean that might exist on a place like Europa?
Yeah. So water is the big deal. Water is the basis of life, and water is one of the big problems of being in space. So for plant biology, like how do you water the plant, right? And so one of the ideas is that just capitalize on the fact that biology is mind-blowingly awesome. There are plants that live in water. And so Artemis 3 is going to be the boots on the moon. So Artemis 1 was a flyby that's already happened. The capsule flying around the moon to show that the technology works was going to be empty. They literally were just going to fly and empty. And so people were like, can we put our stuff in there? And so there are a bunch of experiments that went round, capitalizing on the fact that the capsule was going to go around. There were plant seeds put on that, and they've been brought back and grown up, look perfectly normal. So it's good. The next flight, Artemis 2, is going to be a crewed flight, not with the intention of landing on the moon, but to show that the technology again works and that you can fly around, bring people back, and all that good stuff. Artemis 3 is going to be next set of crew on the surface of the moon. And one of the experiments they're going to take is a thing called LEAF. And LEAF is a little plant growth chamber, and the astronauts are going to take it out, put it on the surface of the moon. I really hope it's a big red button. Press a big red button on the top, right, that starts it, and it will have plants growing in there with lights and stuff like that. The duration of that is probably going to be of the order of 10 to 12 days, just because you know those science fiction movies where the shadow of night comes across and the astronauts running and then they get frozen because the shadow comes across. That's actually really what it's like on the moon. The temperature when the shadow of night comes across drops so dramatically that it just freezes everything. And so the lunar day that we're going to have access to is going to be, it's half of a month. So it's going to be about 15 days, which means that an experiment could probably run for about 10 days. It's going to be plants growing in a chamber physically on the surface of the moon. It's going to be some model research plants, the ones that we work on, this thing, Arabidopsis, Wisconsin fast plants, right? And duckweed, an aquatic plant, to exactly try and deal with this fact that water is both life-giving and an issue, and aquatic plants have solved that for us.
Wow.
Yeah.
Looking forward to that. And that's our Artemis 3.
Yep.
Sometime in the future when that happens.
A few years.
A few years now.
It's
Yep.
You know the classic NASA thing. We all know this. Here's when it's going to occur. No, that's when they said it's going to occur. Add like 4 or 5 years to it. So
Okay.
we're
NASA time.
Yeah, NASA time.
NASA time.
We're Artemis 2 probably next year, something around that kind of thing. Artemis 3 is going to be a few years after.
Fantastic. So I'm going to ask you. So folks, start getting your questions ready. I'm going to ask you guys two more questions. Sort of scientifically, you sort of answered, given a hint of this. Next 5 years, what's the big advance in your relative disciplines?
I think we're going to get close, if not find, an Earth 2.0. We won't know if there's life on it, but I think we already have about 60 exoplanets that are Earth-like. They're all a little bigger than Earth. We're closing in. So I think that's really where it's at in my field in the next 5 years.
Okay.
I think all of our vision at the moment is on the International Space Station. That's a big part of what we do. But everyone is looking at the moon, and the people back on the surface of the moon, finding out what biology does not when you're just orbiting the Earth, just orbiting the Earth, but when you have actually truly moved into that realm, which is kind of like the deep space realm, which is, if we're going to do the voyages of discovery, that's what we have to be able to cope with.
Mm-hmm. Aliens. Yes, no, maybe?
Probably, but we may never know.
Okay.
Play the numbers. Gotta be.
Yeah.
There's
Okay.
just so many stars, so many
could there
planets.
not be?
Yeah. How could there not be? Yeah.
Got it.
But again, distances mind-bogglingly far, like incomprehensibly far. Maybe we would never see them.
Great. Thank you. Who's got a question?
We have
Got
microphones.
mics around? Yes. Well, wait for the mic to come to you.
I'm curious, we know like potatoes from The Martian, but if you were to assemble a sort of starting set of crops to actually take on a sustainable voyage, would it just be potatoes? What would be in that set of seeds?
Oh, French fries. I mean, how could you throw it out? The French fries. So The Martian was actually well thought out, and a lot of it is very, very close to how it's going to be. There's some, I always feel sorry for the people who are watching space movies when we're in the audience, because it's like, fake, there's things which are wrong. The Martian's really good. You want to grow crops which you want to eat most of what you've grown. You don't want a lot of composting and things. So leafy greens are a big one for the moment, and they grow pretty well. Lettuce, cabbage, all of those kind of things. Then things like potatoes actually are target crops. And then the radiation component of it, one of the countermeasures, which is actually works really well, which we all know, fresh fruit and vegetables. Eat your fresh fruit and vegetables. Dietary intake of antioxidants is a very, very good way of protecting biology. And so this is going to sound like it breaks the rules. The rules are you're going to grow things where the amount of food that you get out of it is large relative to the volume of plant that you're going to grow. But the ones that break that are strawberries and blueberries. But that is because the highly pigmented plants are fantastic with antioxidants, and radiation is a big deal. And this is maybe one of the ways to overcome it.
Okay. Yes, over here.
Thank you. Wonderful discussion this evening. I've got a bunch of questions rattling around my head, but I'll try and articulate a couple of the ideas that I had. Was thinking about these like you mentioned, the moon experiment was really cool to me, and then I was like, oh, what a shame that it has to freeze. And then I remembered I have rhubarb that I keep, and it's a plant that it must freeze every season to regenerate. So it's like, I was wondering on that note, and then with like radiation, are there ways to think about engineering maybe certain types of like, if you have a glove of water, why not exploit that you can grow aquatic plants potentially really well? So I don't know if you want to think about like maybe.
Yeah. So I always think that NASA is an engineering organization, and the way that you're going to solve a problem is you're going to build some engineering solution for it. Biology solves things incredibly elegantly, and things like being flooded. Plants naturally get flooded. They have mechanisms that deal with being flooded. So one of our research programs is to take that flooding response pathway and manipulate it to make better flooding-resistant plants. The freezing thing on the moon, the temperatures we're talking about, the coldest place that's been measured is, I think it's the south pole of the moon, physically measured. The temperatures are incredibly low. So they may be moving into that realm where if you think that the reason that biology is good at what it does is it's had millions and millions of years to get good at doing it. And that evolutionary process makes biology phenomenal, but it's made it phenomenal for the Earth. And we're now going to move it into an environment where we may be moving the parameters a little bit too far. But that may be the freezing tolerance may be the way to go. Yeah, absolutely.
Thank you.
Great. Yes. Oh, we got a couple over here.
This is for Sarah, actually. You were talking about looking at other planets and trying to see if there are like biosignatures on them. I think you were talking about doing it visually, or I just want to hear more about like how do you look at other planets so far away without taking a sample and figuring out what that chemistry is from that sample.
Right. So right now our primary tool for this is the James Webb Space Telescope. So we look at a planet where its star's light is passing through its atmosphere, and we take a spectrum. And so that spectrum, different molecules have responses at different wavelengths, and so we can figure out what's there by comparing that to a spectrum taken without the planet there. And so we can figure out then what's in that atmosphere chemically, and from that we can try to piece together how that atmosphere might have formed and what might have caused the molecules that we're seeing in that atmosphere. So it's taken a long time to get to this point, and this is one of the major results out of the James Webb Space Telescope. This is the first time that we can get spectra that are this good, and we can really start to understand the chemistry of exoplanet atmospheres.
There's another hand over here, too.
You mentioned the need for liquid water on the surface of a moon or a planet, but we have a lot of life living deep in rocks, with biogeochemistry being one of the major things in the last 30 years in my experience. How do we look for critters living in rocks without liquid water on the surface?
So all of the Mars rovers actually have drills. So part of the point of that is to be able to dig down deep into the rocks and see what might be subsurface, where it's warm enough that organisms might survive. We don't have a pathway that's been hypothesized to get to life as we know it on Earth without access to liquid water. Liquid water is always part of that process. But if life evolved that doesn't require liquid water, it could definitely be present in those samples. And so we always keep the possibility open, but as far as we understand things at this point with how we think life on Earth formed, liquid water was a key ingredient. There is, it's distributed differently, and so those organisms would access it differently than what we do at the surface. So at the surface, you know, we have oceans and ponds and streams. Microbial life deep in the crust would access that water in a different way. But we actually think on Mars that there may be quite a lot of water subsurface, but it's not at the surface. And so it's only been recently that we've been able to start testing that with the new rovers.
There's no way of identifying that remotely.
No.
You got to get there and dig.
You got to get there and dig and take samples and take measurements.
Yep. Other questions? Yeah, right over here.
So, quick question, but what is the protocol? What happens if and when you do find life?
There supposedly is a protocol. I was at an interesting workshop once where actually Frank Drake was there. It was in honor of him, and they had panels on different topics, and this was one of the panels, and it was led by a federal judge who deals with a lot of FCC related things, and he was talking about the fact that if we received a signal, who does it belong to? The scientist that collects it, the observatory that they use, the funding agency, the government that funds the funding agency that funds the project. Is it something that belongs to all of humankind? So scientifically, people assume that we're all gonna share the news and cooperate, right? It's a scientific discovery. There's a big fear that the military will clamp down on it and cut off communication once it's done. So I'm not sure in the current geopolitical climate what would happen if we actually did make contact. But I know in the scientific world, the assumption is that this would be broadly shared as a scientific discovery, like every other scientific discovery. Anything we do in science that's funded by the federal government should be in the public domain. Taxpayer dollars pay for that, and so taxpayers should have access to that information. So my hope is that it would be in the public domain and everybody would have a piece of that amazing discovery.
It's a great question. So when Europa Clipper goes to Europa and let's say it detects some biological activity,
NASA will have a press conference. I assume. That'll be really good for NASA funding.
Yeah, that'd be
really,
great.
really good for NASA funding.
The alternative is that there's life that's detected and it's kept a secret, and that is a much more efficient government than I think that we have. I just cannot imagine that you could keep that a secret.
Right.
Yeah, it just won't happen. Yeah.
One conference and it's all over.
it's
Right?
all over, right?
Yeah.
I see another hand. I see.
Oh,
Oh,
David.
I don't know.
All right, I'll answer this.
Occasionally you're in an audience and you got like a ringer in the audience. We've got a couple ringers here tonight. So, David.
This is not a nasty question, but it's a genuine question. Yeah, you say we might detect life, but bearing in mind that the odds of fish swimming around on Europa or kelp floating in the oceans or little critters running around on the ice, that's not what we're expecting. Yeah? We're expecting microbial life. Do you think we would detect it if we assume that it may not have any of the familiar biological molecules, and they're not sending out microscopes? They've never sent a microscope on a rover, and as far as I know, don't have plans to. So how do you think we might actually be able to say we have signs of life in our solar system in the next 10, 20 years?
I think if it's Europa, chemically it'll look a lot like bacterial life on Earth. And so we may be able to detect it chemically. Not with Europa Clipper. The chemical instrumentation's not sophisticated enough for that. This is really to figure out how deep the ocean is and how much ice crust is on it and things like that. I think the Titan exploration with Dragonfly will give us a lot more chemical information. And so, there's always that possibility, but Titan's chemistry is really weird. And so we would have to go to the lab and see if we can figure out how, are the molecules that we're seeing actually indicating that there's life or not? And we just don't know enough yet to be able to do that. I think in the end, you're right. We would either have to send something like a microscope or we would have to bring back samples. I don't think there's any other way to know definitively unless we did that.
Plants.
I'm expecting little things crawling around and fish swimming. So I don't. This is not a problem for me, David, at all. I mean, but it is the underlying question is, so if we're expecting it to be microbial-like life, but on Earth multicellularity is a big thing. So why are we only expecting like single-cell microbe-like things when on Earth that clearly is not the only way that biology ends up going? Is there a reason? I mean, apart from the fact that microbes are everywhere and it's like, it seems pretty much like that's probably what we would find.
Right.
But why not multicellular?
I think the assumption is that's where life on Earth started.
Yeah.
And so if we're going to look for the simplest life possible, that's the most likely to find in more places.
Mm-hmm.
But chemically, I don't know. If it's Titan, I have no idea. There are all sorts of very bizarre compounds at the surface of Titan that would be really interesting life to study if we found it.
If life got started microbially, how long did it take to go from single-cell microbes that are hard to detect to something that's multicellular that might be easier to detect?
In the history of Earth, relatively quickly. It did not take long once there were single-celled organisms for multicellular organisms to take off. It took quite a long while to get to the point to see the diversity we see now. Once we got to the Cambrian explosion, that's where life really diversified. But up until that point, going from barren, lifeless Earth to single-celled organisms to multicellular organisms was all relatively fast in the timeline of the history of Earth.
So maybe you're right. Fishes,
Yeah. I mean,
stuff crawling.
yeah, the thing that always is just mind-blowing to me is how soon after the conditions on Earth were like life-supporting from like the origins of the Earth, with all of like volcanic activity and all that kind of stuff, to the point of where there's liquid water, and then how quickly you just get life. It's phenomenal.
And that date for the first life keeps getting pushed further and further back in Earth's history. So it was fast, and maybe it happened many times over.
But I think that's why both of us go, there's
it's gotta be
another
there.
life out there. Because you look at the history of the Earth, it's almost as soon as it could possibly occur. It's remarkable.
Other questions from the audience? Yes, up front. Thank you. We're going to bring a microphone.
Run.
I know that one of the challenges that arises when you're looking for biosignatures or life on other planets is that you might not even know what you're looking for. So if you're not looking for a carbon-based life form, what are the chemical signatures that you might be looking for that could point towards biological activity on another planet?
So right now we're limited to what we can see in the atmospheres. So it's very simple gases like carbon monoxide, carbon dioxide, methane, water, ozone. Many of those are clear-cut signatures of life on Earth. None of the other planets in our solar system have that much water and that much ozone and that much methane in their atmosphere, but they're not exclusively due to life. And so we're having a hard time sorting out a set of things we should look for. And so I think what it's gonna come down to is the ratios between things, because then we'll know, okay, every planet in this zone, habitable zone, might have water and might have methane, but if they're at this ratio, that means that there's something driving it, towards this realm that indicates that there's life there. So there's a lot of work going on in the field right now to figure out what those ratios should be. The challenge we have is every star is different, and so the habitable zone will be different. And so what you would expect to see in a planet in another solar system that is not like our solar system, it could be completely different conditions. And so we kind of have to start over every time we detect a new planet to try to understand, all right, if it's at this location and it's this big and the star's this bright, what could be there? Can we model that? What should we look for? And so we're just now getting to the point where we understand those processes enough that we can start to have an intelligent conversation about what we're seeing. But pretty much right now, every time James Webb looks at a new planet, we get an interesting spectrum, and it's back to the drawing board to try to figure out what it means.
Great. Well, I'm gonna. Oh wait, one more question over here, and then we'll wrap up.
So I kind of circled back to the idea of radiation again. And this is kind of on like the photosynthesis. Like for me, I was like, light is radiation. Plants use radiation to make energy. Are there ways that, and I know certain plants photosynthesize using different spectrum. Are there ways that like plants could adapt to potentially use the higher available energy in space, like from that radiation?
Yeah. So again, it's going to sound super scary. The radiation is not rip your face off radiation levels, but it is going to be damaging, right? So that's where we should. So there's the electromagnetic spectrum, different kinds of radiation. Your cell phone works from radiation. Light is radiation. Biology, the chemicals that make biology work can absorb that energy. And if it's in the right kind of packages, then it will be absorbed and it can be used, and photosynthesis uses that kind of. But that band where the energy is just right, we can call it the Goldilocks zone of radiation, is pretty much the visible spectrum. The reason that you can see things is because packets of energy are hitting your eye and flipping molecules around. If you go to too low an amount of energy, it doesn't interact with biology enough to power stuff very well, at least as far as we know. So that's why none of you can hear the radio frequencies that are running like your cell phone and things like that. Because if you could, imagine like this room will be full of noise, right? But it just doesn't interact with us very well. The radiation, the galactic cosmic radiation, which is the one that we're most worried about for biology, is the other end of the spectrum where the packages of energy are just too big. And rather than absorbing into molecules and vibrating things and making things work, they just smash molecules apart and they cause chemistry. So with how we understand how it works at the moment, that galactic cosmic radiation background is just way, way too energetic for biology to deal with. It has to repair itself rather than use the energy.
So let's wrap up with sort of science is a personal venture, right? We're all individual scientists doing our work in collaboration. So personally, what's on your docket scientifically that you, Simon, will be doing next?
So we're excited about the moon. We actually are beginning to work a little bit with lunar regolith simulants, like for what it's like to potentially use that as ways of growing things on the moon. So we have a lot of questions about how plants interact with the space environment, with microbes and that. But I think all of our sites are going towards the moon. That is going to be, we're already there, but a long time ago, and we're going back, and I think the discoveries we'll make about how biology works are just going to be phenomenal. So moon.
So you've got a lab here on campus.
Yep.
Doing some of this work.
Yep.
A bunch of students in that lab.
Yep.
Okay.
People playing around with lunar regolith simulants. If we are successful, we might even get real Apollo regolith simulants, and then you're going to have to work out, Eric, how to put that into a lab on campus with all of the security that goes with it.
I'm sorry I asked that question. Susanna, how about you?
Yeah, I have two things. First, you mentioned earlier, I'm the provisional director of the Wisconsin Center for Origins Research, or WICOR. We're just launching WICOR this semester. It's a 6-department undertaking where we have collaborative research that spans across science disciplines all over the college, and also some from the College of Agricultural and Life Sciences. But we are trying to understand origin of life, both on Earth and the possibility for life elsewhere in the universe. We have 40 or 50 people involved, and we're really excited to get this launched because it's the first time we've brought together all these different pieces of expertise to work together as a team to try to understand this really complicated problems. So we have astronomers, chemists, geologists, atmospheric scientists, oceanographers, biologists, botanists, And we're all coming together and trying to understand how life may have formed on Earth and whether we're alone in the universe. Beyond that, in my own research group, I have 9 graduate students working in my lab right now. We take spectra of these tiny little molecules we think are the ingredients that lead to biological molecules. And then we compare those to observations using telescopes. And so we've just gotten 2 really exciting grants funded. I've always studied star formation, and now I'm kind of moving in the planetary science direction. I just got one funded to look at chemistry on Titan. So maybe I'll understand, David, how life might exist in that weird chemical environment. And then the other really exciting thing is I'm a partner on a project with the Jet Propulsion Lab at NASA to build a spectrometer on a chip. It's about the size of your cell phone. And if we can get it to work in my lab under the right conditions, we're going to try to send this thing to Mars or Titan or somewhere. So we're really excited about that. We don't think like engineers, so we get to learn from our NASA engineering friends, but we can bring the chemistry. So that'll be a lot of fun.
Great. Well, thank you both. Fantastic. Great work. Thank you all for being here. Thank you. Sam, any announcements we need to make?
Next one is December 3rd.
December 3rd. Come back for more exciting science. Thanks very much. Thanks very much for